My dear students, today with this video I will discuss about the light reaction of photosynthesis. You know, photosynthesis is an anabolic process in which light energy is converted to chemical energy, and the whole process is divided into two phases, light phase and dark phase. Though the nomenclature is like that, light phase and dark phase, but both these phases are performed in daytime. That is, the reactions which are performed by utilizing sunlight are termed as light reactions, and the reactions which are performed in daytime but without the utilization of light are placed in dark phase. Okay, so in the light phase, ATP and NADP are synthesized along with the synthesis of O2, that is oxygen, as a byproduct. And in the dark phase, the carbon dioxide of the atmosphere is trapped and reduced into carbohydrate in the stroma of the chloroplasts. Okay, that is, the light reaction of photosynthesis is performed in the grana of chloroplast to synthesize ATP, NADPH and O2, and the dark phases are performed in the stroma particle of chloroplast to reduce CO2 into carbohydrates. So let us discuss the light reaction of photosynthesis with the help of jet scheme. Here, this one is the jet scheme. Okay, this scheme has been provided by Hill and Bendall, and that is why it is called jet scheme of Hill and Bendall. You see, it is just like this. That is why it is called jet scheme. So what happened to light reactions? I will discuss one by one. First, I will show you, or I will discuss about the photolysis of water. Okay. Here you see, water molecule is broken down into H positive ion and OH negative ion in presence of sunlight. That is why it is called photolysis of water. Okay, so the produced H plus ion, here two H plus ions, they will be kept for the future use. Okay, and what happened to OH negative ion? OH negative ions, they release electrons, and by removing electrons they will be converted into radicals, and the radicals in presence of Cl minus and Mn2 plus will be converted to O2 and H2O. Clear? Again, by means of photolysis of water, water molecule is broken down into H positive ion and OH negative ion. H positive ions are kept for the future use, and OH negative ion, they remove electrons, and by removing electrons they converted into radicals, and these radicals in presence of Cl minus and Mn2 plus converted to O2 and H2O. Now what happened to those electrons? The electrons released from OH minus ion will be transferred to the PS2 when PS2 is in electron deficient condition. Okay. Now the basic question is, what are PS1 and PS2? PS2 and PS1, what are those? These are the pigments. The pigments which absorb the light of wavelength 330 to 680 nanometer are termed as PS2, and the pigments which absorbs the light of wavelength 680 to 700 are termed as PS1, and both of them has photocenters P680 and P700 respectively. That is, the pigments, they trap photon particles and they transfer to the particles to the photocenters of the respective system. Okay. Now what happened to PS2 and PS1? Let us see. Let us consider PS2. Here a symbol of sun is given. That is, the PS2 are absorbing photon particles, and by absorbing these photon particles they will get excited, and excited P680 will release electrons, and these electrons will go to pheophytin and then to cytochrome B6 and F complex via plastoquinone A and B. Again I am saying, by receiving or by getting photon particles, the photosystem 2 get excited, and due to that excitement electrons released, and those electrons are transferred to plastocyanin via plastoquinone, pheophytin, plastoquinone A, plastoquinone B and cytochrome B6F complex. Okay. Then from plastoquinone electrons again transferred to P700 if the P700 is also in electron deficient condition. How? The P700 also receives sunlight, and by getting or by receiving sunlight get excited, and due to this excitement electrons released, that is P700 also in electron deficient condition by receiving sunlight. So that is why, to fulfill the electron deficiency, electrons will be transferred from plastocyanin to the PS1. Okay, you see, during the transfer of electrons from PS2 to PS1, what happened? ATP are synthesized here. That is, ATP is synthesized when the electrons are in the cytochrome B6F complex. Okay, in the complex, in presence of ATPase, ADP and inorganic phosphate combine together to form ATP. Clear? Okay, that is, photophosphorylation has been performed during the transfer of electrons from PS2 to PS1. Okay, now what happened to the electrons which are released from PS1? The electrons released from PS1 is now received by FRS. Okay, ferredoxin reducing substrate. Okay, so these ferredoxin reducing substrate will transfer the received electrons to ferredoxin via few other carriers like iron sulfur proteins. Okay, these are FESX, A and B. Okay, that is, the FD is now in such a condition that it is having electrons. Now it will decide the transfer of electrons in two paths. Okay, and here the paths will be dependent upon the availability of H positive ion. That is, if the light is such that photolysis of water has been performed and H positive ions are available, then electrons will be transferred from ferredoxin to FNR, flavoprotein ferredoxin. That is, FNR is flavoprotein for ferredoxin. And from FNR, electrons will be transferred to this one, NADP. So by receiving electrons, NADP will be converted into NADP minus. These NADP minus will receive H protons. By getting these protons, this one NADP negative will be converted into NADPH plus H plus ion. Clear? Okay. That is, when photolysis of water is happening, PS2 is excited, PS1 is also excited, then the electrons from PS2 is fixed in this form. That is, electron is fixed as NADPH plus H plus ion. Okay, and during this transfer of electrons, that is, the non-cyclic one. Why non-cyclic? Because the electrons released from here never came back to here again. That is why it is non-cyclic process. So in the non-cyclic process, ATP is synthesized. That is why it is called non-cyclic photophosphorylation. Okay. And what happens if the light is such that only PS1 is activated? And in this condition, what happens? In this condition, electrons will be transferred from ferredoxin to cytochrome B6F complex, and during this transfer one ATP molecule is synthesized, and again from this complex electron is transferred to PS1 via plastocyanin. Clear? Okay. And during the transfer of electrons inside the complex, cytochrome complex, what happened? Again another molecule of ATP is synthesized. That is, in case of the light is such that only P700 is activated, then the process is cyclic and two molecules of ATP are synthesized. Here no NADPH plus H plus ion is synthesized. That is, electrons are transferred from FAD to cytochrome complex directly, not to the FNR. And moreover, here in this condition H positive ion is not available, as the photolysis of water is not performed. Clear? Okay. So it is called cyclic photophosphorylation. That is, in case of non-cyclic photophosphorylation, splitting of water is required, one molecule of ATP is synthesized, and at last NADPH plus H plus ion is synthesized. And it is to be noted that oxygen is produced as a byproduct. But in case of cyclic process, only P700 is involved, and here only two molecules of ATP are synthesized. No NADP is synthesized and no need of photolysis of water. Okay. Now what happened to this PS2 when the PS2 is in electron deficient condition? The photolysis of water is resulted into the formation of electrons. These electrons will be continuously transferred to the PS2 when the non-cyclic photophosphorylation is performing. Okay. So this is what about the light reaction of photosynthesis.